Dean, School of Biological Sciences, Central University of Kerala. With over 30 years of teaching and research experience, she has led 7 major funded projects and published around 50 peer-reviewed papers. She is a recipient of Commonwealth Academic Staff Fellowship and the Dr. P.K. Rajan Memorial Excellent Award, among other honors. We are honored to have her chair this session.
Also, it's my pleasure to introduce Professor Govindarao Dudukuri, Professor of Biochemistry and Molecular Biology at Central University of Kerala, and currently the Dean, School of Medical Science and Public Health. His research focuses on inflammation, immunomodulation, cancer, and epigenetics. He has published over 45 research papers and received the Best Teacher and Best Researcher Award from Githam University. He has also served as a head of the department, Dean of School of Biological Sciences, and Director of Research at CUK. I kindly request our respected chairpersons to take the seats on the dais, and I hereby hand over the sessions to the chairpersons.
So we don't need any further introduction about Professor Padmanabhan Balaram sir. So may I invite you, sir, for your keynote address.
We are running late, so let me see how much of time we can really make up. Giving the first lecture at a conference on molecular signaling by someone who isn't really working on molecular signaling is quite a challenge. But I am going to tell you some things that I have learned about molecular signaling while teaching many other kinds of related subjects. It turns out that when you start looking at children's books, right from somewhere near upper KG to class 1, you'll find that picture up on top there, which are the 5 senses, and that's how you begin to teach children the sense of touch, the sense of smell, sight, all of that. And so the 5 senses really determine how we process all the information that you get from the environment. Molecular signaling then would imply a study of the ways in which biological organisms communicate with one another in nature, and also the way in which cells in a complex organism communicate with one another. In some ways, the starting of the society by Professor Shamir Bhattacharya really implies that it has its origins in his interests in the field of endocrinology a long time ago. And endocrinology itself is the study of the ways in which cells in the human body or in animal bodies are communicating with one another over a distance with chemicals, molecules produced by one cell but acting on a target scale. But organisms too also communicate, and that's what I show you here on the first slide. What I illustrate is the classic example, which is the work done, and I will mention this in a moment, by Adolf Butenandt when he discovered the first pheromone. And the first pheromone that he discovered was bombicol, the molecule right in the middle, which is the subject of organic chemistry. But bombicol itself is a molecule which the female silk moth emits. And it emits it so that it can attract the male silk moth before mating. And that's a picture. So you have a biological phenomenon and then you have a consequence. The signal itself here is the molecule. But in order for the molecule to work, it must be recognized by the target, by the male silk moth, and therefore the male silk moth must have a receptor. That receptor is invariably, not always, but invariably a protein molecule. And that protein is also quite often, again not always, a protein molecule which is embedded in a lipid membrane. So it's a membrane-bound receptor. And there you can see the structure of the Bombicol receptor. That then becomes biochemistry or structural biology. That is the receiver. So we have the language now of signals and receivers, so we have the language of communication. And therefore I think a more appropriate title for the talk would be Chemical Communication in Biology, the way in which organisms communicate with one another, with their individual cells in a more complex organism or their individual organisms sort of communicating with one another. So in a way, signaling is across the kingdoms, across all of biology. It involves cell biology, it involves biochemistry, it involves physiology. It also deals with molecules, which is there in the title of your symposium. And therefore, natural product chemistry and structure determination become legitimate subjects of molecular signaling. And today, of course, you can't get away from omics technologies. Now, I won't comment very much on omics technologies, except that it turns out that everything in biology came before the omics technologies. And we are yet now to decide what is it that we have really learned from all the complexities that we get from metabolomics, from proteomics, and from genomics. So in a way, after the keynote lecture, you will be in the position of this little boy. His father asked him, what did you learn on the first day of school? And you'll have to say at the end of my talk, not enough, I have to go back tomorrow and day after tomorrow. There are many metaphors in signaling literature which are used. The most common of them is what molecular biologists and cell biologists use. Protein phosphorylation is really the classic example of signaling that you teach students when you're teaching a course in biochemistry. There you have the metaphors, writers, readers, and erasers. So the writers become the kinases which do post-translational modification of target proteins by phosphorylation. Then the phosphorylases come along and remove the phosphate groups. But in between, the reader now, a protein receptor, actually recognizes the modified molecule. So this is terminology. But the heart then is communication. This communication can be cell to cell, very often hormones, or it can be organisms to organisms. That is pheromones mostly in insects. And remember that everything signals to everything else. And in the field of ecology, where you look at plants, insects, animals all together, they are all talking to one another. And this is where I'd like the students to think about one problem. And that problem is, how do you communicate? The only animal which really communicates entirely by sound are human beings. And therefore, there is the evolution of language. And the evolution of language is a very complex process which follows the evolution of the human brain. So what we call the cognitive revolution is really the development of the human brain, which gives you this ability to communicate with others. Animals don't have this luxury, but they still have to satisfy the major imperatives of biology. They have to survive and they have to reproduce. And they do this very well. You can see that plants which cannot move now are able to attract insects. Pollination is at the heart of biology. And you're recruiting insects now to come and carry pollen grains and distribute them everywhere. So sensory perception and signal transduction are intimately related. But eventually this is nothing but the chemical or physical stimulus acting on a receptor molecule. and resulting now in a cascade of downstream biochemical reactions. It also turns out that the cascade of biochemical reactions that happens is now quite well understood. Today, I don't believe that a great deal of the proteomic technologies which have come, which allow you to pull down all interacting proteins, have really contributed enormously to simplifying and making understanding of the area very much clearer than what it was before. Sensory perception itself, we can recognize molecules. The receptors in your tongue or the receptors in your nose really recognize molecules. But the eye doesn't recognize molecules, the eye actually recognizes photons, and the receptor there is rhodopsin. And therefore it tunes to a narrow spectral region in the visible region. The receptors in your ear, which recognize sound, of course, are recognized now sound waves instead of light waves. And they now tune to a given frequency of sound. And when you touch, you have receptors in your skin which are mechanoreceptors. They are reacting to the pressure. So they're almost like pressure transducers. And all of these are again protein molecules. So proteins are at the heart of all of this. And so what you really need in molecular signaling is that biology or biological organisms must have the ability to biosynthesize a vast diversity of molecules. Look at the number of molecules which are there in biology. I'll ask you that question in a moment. You need to be able to understand their activity and their chemical structures. You need to recognize what kind of effector molecules are recognized by what kind of receptor proteins. So if you have identified insulin as a hormone, you must find the insulin receptor. Afterwards, you must understand the process by which insulin, after binding to the receptor, what is the downstream biochemical effect. These are all the subjects which have been studied now for a long time. And this is high specificity of biological receptor ligand interactions. That's we recognize. At the heart of biochemistry is specificity of molecular interactions. And the study of molecular interactions, as I told you in my introductory remarks, is really the study of chemistry. Right at the beginning, more than a century ago, Emil Fischer had this idea for explaining enzymes. He had this idea of what he called the lock and key hypothesis. So the small ligand molecules fit into receptors just the way locks recognize keys. That is molecular recognition. So the lock and key hypothesis has stood the test of time. All that has happened today is the lock and key. The lock is a little bit flexible, the key is a little bit flexible, and therefore we begin to talk about dynamics of molecules. We have simulations on computers which tell you how molecules are interacting. We get pretty pictures, but nothing fundamentally has changed. Chemistry, as Arthur Kornberg pointed out many years ago, is the lingua franca of the medical and the biological sciences. I showed you this slide yesterday for those students who were here. And you must remember that you can't understand anything in biochemistry unless you understand chemistry. So let's just take a look at Bombicol. I brought this along just to show you because I teach this. And we could ask the question, how did Butenandt actually discover Bombicol? This is what one must go back and read. And the purpose of the next slide that I showed you is that occasionally it's important to go back to the original paper. And never read a review until you have at least tried to read the original paper. And the Bombicol paper, which made Butenandt was already famous because he'd got the Nobel Prize for discovering the human sex hormones and their structures. And here he was working on Bombicol. And then when I went back and looked at this paper in 1959, I found that was the whole paper. That was all. And marvelous, isn't it? The only problem is it's in German. Now, when I was young, you had to study German and translate this, but I've forgotten all my German. And what you do now, you just put it into Google Translate and you get it. But if you put it into Google Translate, it doesn't translate perfectly, so things will come out a little bit jumbled up. But if you know your chemistry, then you can now rewrite the paper again, because your chemistry will get corrected, and after all, you're only worried about chemistry and not worried about grammar at the end of it. So I've translated Buchanan's paper now. And this is what you did, and this is how he found Bombicol. Now it doesn't looks a lot of organic chemistry there, but don't worry about it very much. Just look at this very end over there. How did he isolate Bombicol? He had 500,000 silk moths, and from the glands of 500,000 silk moths, he isolated Bombicol. How much of Bombicol did he isolate? And this was the year 1959. None of the techniques that we use today routinely were available at the time he did this work in the 50s. But he had chromatography, so you could follow a molecule when you were chromatographing it. But how do you follow a molecule? You don't have so much of sophisticated detectors, UV detectors, refractive index detectors, high performance liquid chromatography, none of it. You have classical column chromatography, and you have your eyes. Your eye is a wonderful spectrum photometer. It can recognize the visible region. And therefore, all you need to do if you're now isolating a molecule which is not colored is to make the molecule colored. So how do you make the molecule colored? You have to do some chemistry. So he attached it to this dye here. You can see a nitro-azo group there, and this of course will be colored. The kind of nice colors that you will get in a few days' time at Holi. Everybody will be spraying on everyone else. And he followed the color. At the end of it, from 500,000 glands, he got 6 milligrams. But he had 6 milligrams, but if you look at the molecule, much of the weight of the molecule is the dye which he attached. So now you have to find the structure. How do you find the structure? No spectroscopy available at that time. NMR was in its very early days, and it was very difficult to do it at that time. He did it the classical way. He degraded the molecule. He oxidized it with potassium permanganate and then tried to find out what he got. And he got these simple molecules here. You can see oxalic acid on one side and butyric acid on the other. And he inferred now the structure of Bombicol. Imagine the structure determination. The same thing happened when Andrew Shalley and Roger Gilliman in the 1960s isolated all the pituitary and hypothalamic hormones, work for which they again got the Nobel Prize. They were brutal pieces of work because you needed 100,000 glands, 200,000 glands. You can imagine processing all of this. Today we complain so much about facilities, technologies, and all of this. But this was all work which was done with a minimal amount of current day technologies, and yet this was the kind of understanding that came from this. What did all this that I described? It's chemical analysis. And what does chemical analysis means? Nature produces a mixtures of substances. They're complex libraries of molecules. You must separate them. How would you separate these? You would separate these into individual molecular components by all the chromatographies that you have today. There's hardly anybody who hasn't seen now or works now without an HPLC. And how does HPLC work? It works because of the marvelous reverse phase column materials which are now available. Once you get that, you have more and more machines, you spend more and more money, and then you get structures quicker and quicker. You can ask yourself the question then, how many new and original molecules now come out from all of the surroundings that you have? Lugs. And to sort of wake you up, because we didn't get coffee between the inauguration and the keynote lecture to keep our caffeine levels up a little bit, I wanted to show you, you may think here's an old guy coming along and telling us something that happened in 1959. Now, the old guy is still reading, so you will find that I read this at the beginning of the pandemic. At the beginning of the pandemic, when we had the lockdowns, it turns out that you had nothing to do except to read, because you couldn't go out. And I saw this article in the New Yorker, which somebody sent me on email saying, okay, you read this if you have nothing to do. and this is the title. It said, "Does hurting make us human?" The New Yorker articles are wonderfully written. And what they described was this lady here who is suffering from a strange disease, if you can call it a disease. She had, her negative emotional range was extremely limited. This meant she never felt unhappy. And a wonderful state to be in. She never felt unhappy, and she also never felt pain. And they discovered this when she had orthopedic surgery, and when she came out of anesthesia, and the nurse was ready to give her some oral painkillers to keep her happy, she said she didn't need anything. So the nurse thought that she's being just trying to act brave, and gave it to her nevertheless. But after a little while she discovered that this lady really never felt any pain. And then of course other people were summoned, and she became a subject of investigation. She was completely insensitive to physical pain. If you're completely insensitive to physical pain, that means your pain receptors are not working, something in your brain is not telling you that it's a dangerous thing sometimes, because you could hurt yourself very badly without recognizing that you have done so. What was the problem? And since I was in a hospital in Scotland and all, what would happen? Of course, you can guess immediately that the genomics experts from Oxford and Cambridge were summoned. And once they were summoned, she became the subject of an investigative study. And eventually this paper I traced to the British Journal of Anesthesia, and this is the beginning of the pandemic, so you have lots of time to search the literature, in 2019. And I found that the pain insensitivity was due to this pseudogene. There's a lot of genetics there which is of importance, but it's a person who has a high concentration of a molecule called anandamide. The name is very interesting, anandamide. Then you know that anandamide must be in some way connected to India, and this is anandamide. Look carefully at it. By now I have left organic chemistry. I've come to biochemistry. You can see that it's lipid-like, it's arachidonic acid. So if you take arachidonic acid and ethanolamine and condense them together, you get anandamide. But there's an enzyme called fatty acid amide hydrolase, very important, which now hydrolyzes anandamide to arachidonic acid. So you can imagine a defect in a gene which makes this enzyme, which would mean that your brain would now get high concentrations of anandamide. And anandamide must now be the molecule which is dulling your pain sensations. But which receptor does anandamide work on? Anandamide now is working on the cannabinoid receptor. And the cannabinoid receptor is the receptor which has made hashish and marijuana popular for centuries now. And you can see the origin of cannabis, and cannabis history goes back all the way to 1838, where it's traced down, and only very recently was the cannabinoid receptor really identified well into the 20th century. Now, of course, it's been cloned and everything. In the 1940s, there's a famous article on marijuana by Roger Adams, a very prominent American organic chemist, who was in fact says that Adams repeatedly risked his career and his position, both for his cannabis research and his political ideals, standing up to the forces of intolerance in a paranoid age. When I read this, I was encouraged, because it would appear that the forces of intolerance and paranoid age continue a decade after decade, and nothing's really changed very much. But what are the cannabinoids? The cannabinoids are the molecules which come out, terpenoids, which come out from the cannabis plant, and that's tetrahydrocannabinol. Now, if you look at tetrahydrocannabinol, it was first isolated by this Israeli scientist, Raphael Meshulam, in this paper in 1964, where he did exactly the same kind of work: a plant, crush it up, extract it, purify it, find the structure, and then he decided that these are important molecules. So there's tetrahydrocannabinol, there is anandamide, two entirely different structures, but they now act at the same receptor. But when Meshulam discovered the marijuana's active component, his question was a very interesting one. It's now making you happy, it's dulling your pain, which is why today we have medical marijuana and slowly becoming legalized. Where is it acting? It must be acting on a receptor. It's a signaling molecule now. And if it's acting on a receptor, why is the receptor there in human beings? There's no need for a receptor in human beings. But of course you will understand that it's the plant substances which have had the greatest effects on the human brain. The most important of them, of course, was morphine. And morphine led to the opioid receptor. And marijuana led to the cannabinoid receptor. So it turns out that these are subjects which are of some interest to the narcotics enforcement agencies, but they are of great interest in asking the question, why do human brains recognize plant-derived molecules? Evolution doesn't make any sense. There must be an endogenous substance. That question, what is the endogenous substance which is recognized by the brain receptors? That's what led to the discovery of the enkephalins and endorphins, which are the substances which bind to the opioid receptor. And the entire field of neuropeptides, the entire field of recognition of molecules by receptors, really exploded in the 1970s with the discovery of the enkephalins and the endorphins. Meshulam was following this, and what Meshulam asked the question was, what is in the brain that is similar to tetrahydrocannabinol? So what did he do now? 1964, he persisted. In Science in 1992, nearly 3 decades later, he published another paper. And this paper was, he took pig brain and again mashed it up and searched for a molecule, an endogenous molecule. He discovered anandamide. And he had same problems. 0.6 milligrams from 4.5 kg of porcine brain. The reason I'm telling you this is only for one reason. Never underestimate the technicalities of doing an experiment. Can you imagine processing 4.5 kilograms of brain and then trying to get from it 6.6 milligrams of a pure substance? That's heroic. And once you've done that, you've really made a discovery. What is the biological assay? This here illustrates chemistry and biochemistry. This is nothing but the classical radio-labeled ligand which the biochemists have been using for a long time, ever since Rosalind Yallow and Burson discovered these assays. You have a labeled ligand and displacement of the labeled ligand, count radioactivity by the natural ligand. That's electron impact mass spectrometry, very primitive now. That's how he got anandamide. After he did all of this work, of course people now like this work only because it was related to marijuana at that time. And so he had a press conference. And when he had a press conference, he was asked, how did you choose this name anandamide? He's an Israeli scientist, and it's coined from a Sanskrit word, which of course means bliss. And he said, why didn't you use a Hebrew term? And his answer is very interesting. He says, we looked for a Hebrew name, but as you may well be aware, Jews are not very happy. We have a lot of words for being down and so on, but not so many words for extreme joy. And this, of course, tells you the condition of many parts of the world for a very long period of time. Science sometimes can also teach you history. Today, the biosynthesis of the cannabinoids has been worked out, but we can return to arachidonic acid. There it is, anandamide, arachidonic acid, ethanolamine. This is just a small digression for the students. This is how you begin when you do the biosynthesis of arachidonic acid. You have stearic acid and you have to convert it into oleic acid. Look at the structures. The structures are such that right here, in the middle over here, you have to remove 2 hydrogen atoms. If you were a physicist, you would of course remove the two hydrogen atoms by having a Maxwellian demon come along and pull out the two hydrogen atoms. If you were a chemist, you would throw up your hands and say, there's no way of distinguishing those two methylene groups from every other methylene group in the molecule. But still there's an enzyme. This is what evolution has really done. An enzyme has evolved to do this, and many years ago, Konrad Bloch actually wrote this in 1969. He would say that this kind of reaction is such that it's predictable in principle, but this would seem to approach the limits of the discriminatory power of enzymes. We have become so used to biological specificity that we don't even wonder about it. We must wonder about biological specificity. It is so remarkably precise. All of modern research depends on methods. And since it depends on methods, it's useful to go back to the founder of chromatography, Mikhail Schwedt. What he said there was, an essential condition for all fruitful research is to have at one's disposal a satisfactory technique. Then he quoted from Descartes. He said, all scientific progress is progress in a method. Everything that you do today, when you get your sophisticated instrumentation centers, you immediately know that the instruments there are sophisticated. But you need so many instruments that you will eventually find that nobody is a specialist in any one method. And since you are not a specialist in any one method, and particularly a sophisticated method, there's a great danger in the kind of interpretations that one makes. Today, my major worry with the way we are going into omics technologies is really this, that the omics technologies really allow you to study thousands of molecules at the same time. What I've illustrated here is one molecule at a time, and one molecule at a time has given us these results. So when you find that 1,000 genes are being upregulated and 1,000 genes are being downregulated, the question really is, what conclusion are you going to draw? And you must think about these things. These are the techniques which have come over the years, decades: crystallography, NMR spectroscopy, mass spectrometry. Every one of the techniques has its roots in physics. The original discovery of X-rays and the diffraction of X-rays from crystals. In the case of NMR spectroscopy, the fact that nuclei, atomic nuclei, have magnetic moments. Rabi wanted to measure the nuclear magnetic moment. In the case of mass spectrometry, again, gas phase measurement, descending from J.J. Thomson's determination of the mass to charge ratio of an electron. So physics cannot be ignored completely when you think about the techniques, neither can chemistry. But coming back to biochemistry. Biochemistry in nature can happen at remarkable conditions. This is biochemistry at 100 degrees. This beetle here, the bombardier beetle, is often harassed by ants. And when it's harassed by ants, what it does is it just throws out a flame. It's a biological flamethrower. It throws out the flame and incinerates the ants. Marvelous procedure. It has an organ which does this, but the chemical reaction which happens really, you can see, is the quinone, hydroquinone reaction, which produces oxygen, which then combusts. This phenomenon was first observed and a paper appeared in 1969. Now today, if I read this paper, that's the year I started my PhD. I've always wondered if I had read this paper when I was about to start my PhD, maybe I would have been fascinated by this and entirely gone into another field. Sometimes it is important to go into a field which is just in its infancy, not to a field which is famous and mature. Tom Eisner's work in this is led to the field of chemical ecology. And chemical ecology, he describes so wonderfully here, is the product of a partnership between biologists and natural product chemists, united by a shared vision and empowered by complementary skills. This vision stems from the realization that all organisms emit chemical signals and that all in their respective ways respond to the chemical emissions of others. There can't be a better quote to talk about molecular signals which are used in molecular signaling. It's all over biology. All these molecules are what biochemists call secondary metabolites. And the one thing that is in biochemistry which is a misnomer is nothing is secondary. The plant invests an enormous amount of metabolic energy in making these molecules. And when it does that, there's nothing secondary. In fact, I would say it's the primary metabolites which are sort of routine. It's the secondary metabolites which do all the interactions or signaling to the outside world. An old review on this subject called Secondary Metabolism represents the splendid idiosyncratic diversity of nature, endowing different species with specific solutions to biological problems. You can't say it better than this. Look at the alkaloids. No better place to look at the alkaloids than Kerala. Caffeine. I don't think we should worry about cocaine and morphine, but there's a fair amount of nicotine which people use, a fair amount of caffeine which everybody uses, and there's a reasonable amount of the cocoa plant too. All kinds of subjects come here: organic chemistry, stereochemistry, molecules, one molecule produced by one plant which will have the coriander, the other one would have the lavender, and there isn't much difference. Well, look at this. And again, no better place than Kerala to show this. You look at the spices, you have all these molecules. You can look at the similarities. You recognize spices immediately once you put it on your tongue. That's molecular signaling. What are the molecules? There are the molecules. Receptors are in your tongue. Signals going on. Now we know a few years back the TRP channel, or what is called the transient receptor potential channels, they were the ones which were recognized with a Nobel Prize for touch and smell and so on. Curcumin, capsaicin, all these have similarities. You can see the vanilloid skeleton here. But look how much trouble nature takes to make these. This is the biosynthesis of capsaicin. So many reactions. Each arrow needs a catalyst. That's an enzyme. That means there must be a protein. If there's a protein, there must be a gene. And if this is how all of biochemistry works, substrate to product with an enzyme, and the enzyme must have a gene, otherwise it can't be produced. And if you have a biosynthetic pathway, all these genes must be lined up in an operon so that you can start them all off at the same time. You must control the synthesis. It's absolutely mind-boggling. Today, how many molecules does this plant produce? The genus itself contains 50,000 varieties of pepper. And there are so many molecules which come out as the major secondary metabolites of pepper. And so there's a great deal to be investigated. But the real high point of chemical ecology is this, which tells you how signaling is used in nature. The chemistry of sexual selection, in the case of this moth. And in the case of this moth, for the moth to reproduce, it must use an alkaloid from a plant. So what it does is, it does this remarkable thing, that this plant produces this alkaloid. The male moth now goes and extracts the alkaloid from the plant and processes it. It processes it, and you can recognize the chemistry here. That lower part of the molecule looks like that. It then processes it to give that molecule, and that molecule now it emits outside, that attracts the female moth, they mate, and when they mate, it puts this molecule into the eggs itself. The result of this is that this transfer of information, this molecule is protective, and therefore it protects the offspring. So here there is attraction of the mate, protection of the offspring, all using a chemical which is produced by a plant. So now the plant and the moth cannot live without one another, really. Now you might ask, why is the plant producing these molecules? These molecules are invariably bitter. You can try, you can go outside, take one leaf, chew it, and you'll feel like spitting it out. All the alkaloids are bitter. It prevents herbivores from coming and going. We are all herbivores, but we cultivate plants and kill them, and nature didn't really intend it that way. So you might ask, as far as chemistry is concerned, how many chemicals are produced in nature? That's chemical space. You can ask how many living organisms are there in nature? That is biological space. And then you can ask, how are these chemicals synthesized? Why are these chemicals necessary for the organism? How are the chemicals of one organism recognized by the target organisms, that is receptors, the subject of molecular signaling? The estimates for biological organisms keeps on varying. We are always protecting our biodiversity, but we don't know any number for what we are protecting. We are destroying, protecting, all of that's happening at the same time. E.O. Wilson suggested 1.5 million species in 1992, but the species definition itself in biology sort of breaks down when you go to microorganisms. Chemical space estimated at about 1 million in the case of natural products, a large number of molecules. And so you have all these fields which are now developing, all of them related to signaling. How do you even estimate how many species there are? This is a very interesting paper. Those of you who are interested mathematically might look at this. The title of this paper, which attracted my attention, sent to me by a colleague in statistics, is How Many Words Did Shakespeare Know? We know how many words he used, but we don't know how many words he really knew, which he didn't use in all his plays. You could ask this of any author. If you go to the internet and ask this question, you will find this question's been asked for Harry Potter, it's been asked for Charles Dickens, it's been asked for all the people who wrote in English, how many words did they know? And you estimate it from the number of words that they used. How many words did they use multiple times? And there will be some words they used only once, twice, thrice, something else which they use 100 times, and so on. That's how ecologists estimate the number of unseen species. Chemistry itself, you can see how it's growing, the number of molecules, because there is a registry, the Chemical Abstracts Registry, which shows this. It's exploding. It's rather like the human population. So centrally in biology, we try to connect genetics and evolution through chemistry. And in conclusion, I have only one justification for accepting to come and speak in a conference on molecular signaling. One of them, of course, is the attraction of coming to Kasaragod, which I have not come to before. But the other one is that I was, in fact, and still do occasionally, whenever I am able to, follow a project which we started on marine cone snail venom, which contains hundreds of peptides. And the advantage with all these peptides is venom peptides act on membrane receptors in channels, and therefore membrane receptors in channels, you have hundreds of molecules, in fact thousands of molecules, you have lots of receptors, so lots of things to study. A problem was introduced to me by Professor K.S. Krishnan, who actually was from Palghat, and therefore he used to go around the southern coast of India and collect snails. He would give me the venom ducts or the snails sometimes themselves, and then we'd mash them up and study. So that's what I was doing. And I'd use mass spectrometry to study them, and this was the reason why we were studying them. Now, of course, what do venomous animals do? They produce venom, they act on target receptors, they immobilize and paralyze prey, and then they swallow the prey. So fishes, worms, all of these are targets of the cone snail. They are paralytic toxins, therefore they are of interest because they will also act on human channels. But the predator-prey relationship is a classic problem in biology. The predators evolve new weapons and the prey evolve new defensive weapons. It's exactly like it is the biological arms race, exactly parallels the arms race that you see, say, Russia and America, or now Iran and America sort of evolve. If one has one weapon, the other one would like to develop another weapon to counter it. That's what biology does. And so there's this constant evolution. And so there's a hypothesis which the ecologists use, which I will show you. Because biology has some very strange things. Like we isolated a molecule, and we also sort of sequenced using transcriptomic analysis the gene for the molecule. And then we found that the same gene, which we got from the cone snail, is also present in the wings of the monarch butterfly. Now you can't, for the life of me, I couldn't see any connection between a marine cone snail and a monarch butterfly. But there must be some distant connection between these. And the reason advanced in this paper, which appeared in PNAS, was that this is because if you have this molecule, the predator doesn't come and bite the wings of the monarch butterfly and completely immobilize it and later on kill it. So it has evolved this molecule. Where it got it from, I do not know. But the red queen hypothesis is something that you should know as students. The red queen hypothesis is from Through the Looking Glass, which is Lewis Carroll's sequel to Alice in Wonderland. And my entire career has been built by reading Alice in Wonderland and Through the Looking Glass repeatedly, because they are full of gems of science. And Alice will meet the Red Queen, and they'll be on this chessboard. They'll see the chess piece, and they'll play a game in which Alice will run around. And after some time, Alice is a very keen observer. She looks around and tells the Red Queen, we've been running for a long time, but we seem to be in the same place. And the Red Queen, who's a typical professor, now says, she's describing the looking glass world. She says, now here you see, it takes all the running you can do to keep in the same place. So many times in research, you will keep on running, but you will end up actually in pretty much the same place. But as far as toxins are concerned, this is what it means, that the predator toxins and the prey receptors evolve over time. But what is interesting is, the more they change, the more their interactions remain the same. Because finally, all the interactions between molecules, small and large, small and small, large and large, they are interactions between atoms. And remember one thing, that the interaction between atoms are governed by the laws of physics and chemistry. And the laws of physics and chemistry are really immutable laws. They can't be subject now to mutation and change. So finally, as a sort of nod to my origins in chemistry, I must ask the question, who are nature's best chemists? So some years ago, I made this slide, and I thought microorganisms are nature's best chemists because they now make the most remarkable molecules, because they survive the most tremendously diverse environments, under the sea, high temperatures, low temperatures, high pressures, whatever. And so Carl Lewis had just passed away, the man who discovered archaea and gave us really the tree of life. And I found this tribute to him on the internet, which says, nature is to be found in her entirety, nowhere more than in her smallest creatures. And I could paraphrase this to say, chemistry is to be found in its entirety only in nature's smallest creatures, that is, in microbiology. But I made this slide maybe 10 years ago, into the early days of my retirement. Now, after I've reflected for a decade in my retirement, I've come to the conclusion that I must change this, but I haven't yet changed it. And I must say, today I believe plants are in fact the best chemists. The reason for this, in retrospect, seems obvious. If biology's primary purpose is to survive and survive until you reproduce, then any organism which is unable to move has the greatest problem in surviving. And it also has the greatest problem in reproducing because it's unable to have any motion whatsoever. The result of this is that plants now have developed ingenious ways and ingenious chemistry, both for protecting themselves and also for reproduction. And so the imperatives of biology dictate that we must take chemistry seriously, and that both microbial chemistry and plant chemistry, there's a huge amount of work still to be done. I hope that you will have a most enjoyable conference, and at the end of it, many new ideas will come to you. Thank you.
Thank you very much, sir, for your nice talk on chemical communication in biology, encompassing the interface between chemistry, biochemistry, and signal transduction, involving both in exogenous as well as endogenous ligands and also receptors like anandamide and cannabinoids, and both molecules or both ligands bind with the same receptor and also induces the signal transduction. Wonderful talk, sir. Now the session is open for discussion. Any questions from the audience, please? Sir, may I know, here, sir, may I know actually the evolutionary perspective of the receptors or the having the biological or chemical diversity? So what is your perspective of signal transduction in terms of receptors? Why I am asking this question is because, for example, take HIV. So it targets CD4 receptor. And CD4 receptor obviously is one of the important receptor for T helper cell activation and for induction of the immune response. So how pathogens evolved to target the receptors so as to influence the consequent signal transduction and also to make use of the host cell machinery for their sustenance, pathogen sustenance. Is it the receptors which are cardinal for pathogens to make use of for their sustenance and for killing the target cells?
Yeah, I think the question that you ask, rather than take the example of HIV, I'll take a more recent example, which everybody should be still familiar with, is fast receding from memory, and that's the coronavirus. Now, the coronavirus, which is the SARS-CoV-2 virus, enters human hosts through the ACE2 receptor. And now you can ask yourself the question, how long have coronaviruses been around? There were coronaviruses before this one came along. So if you look at common cold, about 30% of common cold is coronaviruses which have been known for a long time. So the first one was a virus which has the name of 229E, and then there are others with other names, OC43 and things like this. These coronaviruses were isolated from the nasal secretions of people who had colds, classically in the 1960s. And there have been veterinary coronaviruses in chickens and so on. So coronaviruses are known. They have been acting. Which receptor did they act on? It turns out that they all acted on different receptors. Viruses use protein molecules on the surface of cells, and it is really a random event. It turns out that in evolution, everything is a random event. Life itself is a random event, the origin. So change, accidental happenings, and if that accidental happening is now beneficial, that now propagates. So you now realize that leucine aminopeptidase on the surface of cells is a receptor for 29E. Angiotensin converting enzyme is a receptor for the coronavirus, SARS-CoV-2. The result is that these are opportunistic viruses. If they find a target, they will jump. The whole question in the coronavirus case was whether any part of the virus had now been engineered such that instead of targeting, say, a different animal cell, it's now targeting humans. So today in the laboratory, you can humanize viruses so that they target human receptors. So that's an evolutionary process. It's rather similar to the predator-prey things. It's constantly evolving. And I'll tell you what my take on the coronavirus pandemic is. You can look back on the coronavirus pandemic, and I think this is how you will look if you look at all the mutations that people have catalogued over the last 4 or 5 years. It's an evolutionary experiment. Who conducted the experiment? Nature conducted the experiment. If nature conducted the experiment, the subject animal was, of course, all of us. And this is the only animal experiment that I know in which the animals are analyzing the results of the experiment and not someone else. The result of this is that it's an experiment in natural evolution. The virus has no advantage in killing all the hosts. The virus is carrying out a process of selection. All of us who still survived the pandemic have now equilibrated with the virus. The virus has equilibrated with us, and now we will live happily ever after until the next virus comes along. This is the state. Our genomes contain all the evidences for interaction of ancient viruses with some of which have even integrated with the genome.
Yeah. Dr. Balaram, this is Vijay. I really appreciated your talk, and it was very inspiring. You know, the basis of chemistry and biology. So I had a trivial question, and I have also been interested in the cannabinoid receptor for a strange reason. It turns out the cannabinoid receptor are also present in human sperm and sea urchin sperm. I have no idea why they are there. And the question was, I don't know if it's known why plants make cannabis. Why plants make cannabis? I don't think I had an answer to that, but essentially what I wanted to say was your presentation was very inspiring.
I must honestly tell you, I haven't seen a good reason why plants make cannabis, but it turns out that many molecules we don't really know what is the origin of the molecule. See, many times some of them are to really function as antimicrobials, I think either for fungal infections or for bacterial infections of plants. It's a terpenoid also, it's not an alkaloid. So you don't really know. Occasionally molecules have been found which now are used by the plant to signal to another organism to help it in some way. We haven't yet understood all the interactions between all the plants and all the microorganisms in the environment. See, there are so many beneficial bacteria which plants actually accumulate. These now are used as make antifungal agents which prevent the plant from succumbing into fungal infections. So there may be other functions in the plant. Yeah.
Plants make this. I had a very stupid reason why plants make this. Maybe they are selfish to make humans propagate them. So.
Most things are more ancient than humans in biology. Humans have come along rather recently.
No, I could not keep quiet because I teach secondary metabolism. And in the first class, I emphasize to the students so that they get interested in listening. Otherwise, it's so boring. All light, a lot of chemistry. No, in their turn, not me. Okay. The reason is that is like a chemistry. A library of chemistry they keep. You never know which one will become useful. And a very general benefit one can think of is that they try to repel when they want. If they want something to come, they attract. So this repelling and protection, they do it against all kinds of people, including humans.
I think Professor Raghavendra makes a very important point. If you look at plant biochemistry itself, a question has been asked. It turns out that enzymes in plants are in fact more promiscuous in their choice of substrates. And therefore they can do the same chemical reaction on many substrates and generate a diversity of molecules. This generating molecular diversity is very important. It is biology's shotgun approach towards something that might be inimical to the organism itself in future. Absolutely right. It's overkill sometimes in terms of biosynthesis, but it's probably metabolic energy which is well invested. Yes. Yeah.
Good afternoon, Professor Balaram. I am Professor S.S. Bhattacharya from Tejpur University. I am also being very much intrigued by your talk, and especially that secondary metabolites part, because I worked with capsaicin and capsaicin of the chili. And the chili plant, if you grow the chili plant species endemic to some place, say like Kerala, which is hot here, and if you take that to the Bangalore, it will not be again hot. So that is because the capsaicin changes, capsaicin upregulation changes depending on the soil. So are we not ignoring the substrate upon which the plants are growing rather than only focusing on the secondary metabolites?
Yeah, this is also absolutely right. Today, for example, plants are more complicated, but if you look at microbial metabolites, it turns out that if you now put on a Petri plate an organism whose genome's been done, there is no evidence that the organism produces those kinds of metabolites, but you can recognize the genes which result in that secondary metabolite production. You can now ask what induces those genes. Now it turns out that if you have another antagonistic organism you're able to find, then you turn on these genes, which then means that they are cryptic metabolites. So the ability to biosynthesize them is already there in the genome. It's a little bit like the example that I discussed yesterday in human evolution. It's a little bit like how adaptation takes place. You have abilities which for one reason or the other you don't use. Like for example, our abilities, humans' abilities to digest milk is really by lactase persistence. So that gene now persists in the populations which are happily drinking milk. But biology didn't make mammals to drink mother's milk after weaning. But the ability to make that enzyme was there, but regulation shut it off after a few months of fetal development. So this is all over the place. But in the case of plants, there's an even greater complication. That is, when you go from one soil to the other, the stimuli that they get from the soil is completely different because the soil is teeming with microorganisms. And today, for example, root microbiology is a very important place to go because that's where many, many wonderful organisms are there.
Yeah, thank you very much, sir. Owing to paucity of the time, so if your audience has any questions, please restrict your questions at the time of tea time. Thank you very much, sir.
Thank you, sir. Thank you, Dr. Padmanabhan, for the insightful keynote address. We extend our gratitude to chairpersons of the session, Professor Sudha Kapali and Professor Govind Rao D, for your time and coordination.
We shall now pause for a short coffee break. Kindly reassemble after 5 minutes to continue the session. Please not